Hi everyone! Tonight's video is on the rough endoplasmic reticulum, which is abbreviated the RER, and we often just refer to it as the RER, so you don't need to learn how to spell rough endoplasmic reticulum. So the rough endoplasmic reticulum, it is a series of membranes that are continuous with the nuclear membrane. If you look at the picture here, this series of membranes here is the rough endoplasmic reticulum. And you can see they're attached to the nuclear membrane. So it's all comes together, and coming out to the cytoplasm is this series of folded membranes. The reason it's called the rough endoplasmic reticulum is that originally microscopists could look at it and see there were dots on it. It looked rough. It didn't look smooth. It turns out those dots are ribosomes, and you should have already watched the video on ribosomes. So the function of the rough endoplasmic reticulum, there are two main things that it does. First of all, proteins that are going to be either exported from the cell or placed inside a membrane are made in the rough endoplasmic reticulum. The next thing that the rough endoplasmic reticulum does is it assembles new membranes, and it assembles them from phospholipids that are made in the smooth endoplasmic reticulum and with proteins that are made in the RER. So we can actually see the rough endoplasmic reticulum using an electron microscope. If you look at these pictures here, this would be the nucleus of the cell, and these dark areas are where the DNA, the chromatin is. But you can see outside the nucleus are these folds of membranes. They look like flat sacs. That's the RER. And at a higher magnification, if you look here, you can just about make out the dots on the outside of that RER. Those are the ribosomes attached to the RER. So how does the RER work to make proteins that are going to be exported from the cell? Well, you remember from the nucleus video that the nucleus contains the DNA, and the DNA never leaves the nucleus. So in order to get the instructions for how to make a protein out to the cytoplasm, a copy is made in the form of messenger RNA, and the messenger RNA leaves the nucleus through a nuclear pore, and it goes out to the cytoplasm where a ribosome can read the instructions in the mRNA and start assembling amino acids into protein. If I then expand this view, so here's the nucleus up here with the nuclear membrane, and then expanding down here is the rough endoplasmic reticulum. Here is our ribosome reading the mRNA in the cytoplasm. And it's beginning to make this protein. It's beginning to assemble the amino acids into the protein. And then all of a sudden, it will come to a signal in the mRNA. There'll be a small message there that tells the ribosome, no, wait a minute. This protein is going to be exported from the cell. You need to go to the rough endoplasmic reticulum to finish making this protein. So the ribosome will then bring this mRNA to the rough endoplasmic reticulum and actually dock on the rough endoplasmic reticulum. At this point, the protein that the ribosome is currently making gets inserted inside the lumen. The lumen means the interior of the RER. So now the ribosome continues reading the mRNA, which is in the cytoplasm, and it's kind of inserting the protein as it's being made. Now, proteins fold up into their secondary and tertiary structure as soon as they're made. As soon as they're made, those interactions between the backbone form the secondary structure. The interactions between the R groups start forming the tertiary structure. So you'll see that proteins are already folded up as soon as they are made. And the RER has many, many ribosomes docked onto it, each one making a protein that is either going to be inserted into a membrane or exported from the cell. Once that protein is completed, the ribosome will then let go of the mRNA. It actually comes apart into its two subunits. The mRNA can float away, and another ribosome can come along and read the instructions and make more of that protein. Now, this protein is not quite done. Once it's fully assembled, all the amino acids have been assembled, there's another step that happens inside the rough endoplasmic reticulum. And that is that sugars are added to the ends of this protein, making it a glycoprotein. These sugars acts as an address label, to tell the RER where is this protein going to go next. Because it's going to be exported from the cell, it actually has quite a long journey before it's going to be squirted out of the cell in another process called exocytosis. So the RER makes proteins that are going to be secreted from the cell or inserted into the membrane. It also puts glucose units on these to give them address unit labels so they know where to go next. And then the process I haven't diagrammed here, but that the RER is responsible for, is assembling phospholipids made in the SER and proteins made in the RER into new membrane. That's all for tonight. We'll see you in class tomorrow.